I get the afternoon post-lunch slot, so I'll bring the energy here. Thank you, everyone, for making the time. It's really, really good to be here. What a morning. You know, I've been following Recursion for a while. I'll tell you a little bit about me. But it was just great to see the constant evolution that's been happening from the presentations from Chris, Lena, Laura, and much more to come. But given this is the first time we're meeting, I thought I'd spend a little bit of time telling you a bit about myself. So I've been at J&J, and I'm joining Recursion from J&J for 6 years. At J&J, I headed up the pharma portfolio across oncology, immunology, neuroscience, and more, as well as being their chief data science officer. So I built up the data science team from scratch there. So you can imagine, and you'll hear this quite a bit when I'm speaking, the convergence of the two disciplines, pharma, making medicines, and leveraging AI and technology to do it in a better way is something that's going to be consistent in many things that I say today. My background prior to that, PhD in organic chemistry. Yes, I love pain. I also, when it was not cool, a background in computer science and a lot of business background as well. And I think the convergence of the three is something you're hearing a lot in the conversations today, which is it's important not to just to have good science and technology, but to actually make sure it's outcomes focused. So that pragmatic and disciplined approach, I think, is going to be really important from what I see in the space. So with that preamble, I'm going to focus the next few minutes, on 3 questions that I get way too often. First question, why AI in healthcare? Although I have to say, I'm getting that question less. So I'm going to spend a little bit of time. The second question is, what can AI do in the life sciences space? What's real? What's not? What is it going to take to get us to the next level? And then the third question that I've been getting a lot in the last month and a half is, why Recursion? So I'll tell you a little bit about why, and also what are the areas I think we need to do more, because it's a constant evolution to be the best. So with that, let's get going. So as you'll get to know me a little bit, you'll see all my presentations will start and end with the patient in mind. I just got back from a trip in Singapore where I was with my mom for a few weeks. Really, really frustrating time. She has Parkinson's and now dementia. And you just get the runaround from physicians because no one really understands the biology of why you get dementia. And quite frankly, it's not even one disease. So how are you going to treat something when you don't even understand the first order principles well? And when people ask me, why do we need AI in healthcare? The answer is simple, right? Look at all the patients that are waiting. And on top of that, you can add stats to that, right? 10% of the drugs fail. 10% of the drugs succeed. 90% of the drugs fail. But if you go a little bit deeper, across the healthcare space, there are many areas where you can actually have impact, and we're starting to see that using AI. And a lot of you know this, right? But I think the one part that gets not the attention that it needs is really around better understanding disease causality. Again, if you don't understand what problem you're solving for, how are you going to do it in a better way? Now, there has been progress with medicines and genetics. Some of you saw Alnylam's result, very good to see with RNA therapeutics, but there's so much more to be done. And if you can understand that leveraging multimodal data, then you get to a point where you can actually diagnose disease earlier. I mean, for today, 40 to 50% of cancers get diagnosed too late. So no matter what drug you have, the outcomes are impacted because you're not getting to the patients early enough. And then the next 2 areas where I'm going to spend more time on is really around developing molecules. Let's think about this for a second. The chemical space, if you're making small molecules, that's 10 to the 60th. I'm a beach person. And I've heard this analogy before, so it's not mine, not an original one. But there are more molecules, and the chemical space is bigger than the number of grains on a beach in the entire world. So that is a big problem to solve for. And that's where you need AI, you need compute, you need scalability, and so forth. Same thing in biology, trillions of cells. You need to understand that better using AI, using compute. But it's not just enough to actually make a good molecule, as many of you know. Therapeutic development is a huge area where a lot of molecules fail. And to me, that's almost the last mile in terms of ensuring that we're actually designing the programs the right way, that the molecules have the right PK/PD. You heard a little bit about some of the algorithms, supremely important to do that better. And then the super last mile, which is actually ensuring the medicines get to patients. This is where personalization, access, and adoption is huge. And last but not the least, we talk a lot about productivity. It just makes sense to do more with less and then improve the quality of care by doing so. But let's zoom in a little bit more in terms of the life sciences space, which is why we are here today. So we're going from a world of, again, as I mentioned, genomics, there's been some great strides, RNA therapeutics, some MD simulations that have cracked the code for some hard molecules, but it's still not enough. And then if you look in development, and this goes back to the example I used earlier, the imprecise disease definitions, how many diseases today are named after either the physician that identified the disease, or it's just a broad description of the disease. It's not based on the biology that's driving the disease. So it's really hard to get to the right patient selection if you don't understand what's causing the problem. So look, there's a lot to be done here, and I'll go through it more in detail, but multi-omics data is creating a massive difference in better understanding not just the target, but if you jump into development, being able to stratify your patients better so that you can actually realize the promise of precision medicine. And why do you do it? You want to take the V, which Laura mentioned, and you want to turn it into a T. So better predictions up front, pull that through in development, and ensure that you do it better, faster. Sounds easy, not really. I mean, if there's something that's humbling in my entire career has been making medicines. It's really complicated. So then let's get to the second question that I get all the time. Where are we today with AI? A lot of you have seen the S-curve. We're not all the way at the top. We're not all the way at the bottom. We're at a really interesting point. I would call it the pre-tipping point. I don't think we're at the tipping point yet. And the reason for that is the following. Some of the examples here, just listed on the left-hand side, are great areas of progress. Like AlphaFold, I mean, as a chemist who was actually also leveraging computer science to be able to predict the protein folding and then find something that would bind and ensure that that doesn't get chewed up immediately before you even do any experiments, it's close to my heart. Huge amount of progress made there. Also, in terms of how do we better execute our trials, ensuring that there's greater diversity. Some of the prior work that I did was focused on that. You know, seeing upwards of 2 to 3x increase in enrollment rates, getting diverse patients faster because you are actually going to where the patients are at. A lot of publications, of course, from the FDA in terms of guidance as to how do we actually leverage AI in drug discovery and development. All that's good, and also some great examples from the work that Recursion has done, looking at molecules, this is during COVID, that exist in the real world, and using phenomics to be able to anticipate which ones could be effective. But here's one problem. You heard themes of this. A lot of these are really important, specific point solutions. But in order to actually make a molecule and then turn it into a medicine and get it to the patient, there are multiple steps involved. Can't fall in love with an algorithm. Fall in love with the outcome, which for us is to make medicines for patients. Next point, which I think is extremely important, and I've seen this be challenging sometimes, which is then is the exuberance of, we need to fix it all. The cost to change and pioneer in this space is high. So to prioritize where you focus, where you innovate, where it matters the most, it's extremely important to be smart about it, and I'll talk more about that. The third, the amount, I think somebody mentioned at the fireside chat that whether it's pharma and whether it's biotech, the adoption hasn't been what it needs to be. And one of the big reasons for that is some of these solutions are not as democratized yet. It's true. But the other is culture and agility. Because if you don't know what good looks like in your organization, it's really hard to cut through the hype curve. And I think that's one area which I call it the bilingual talent and culture. Is crucial to the companies that not only you want to work on the right problems, you want to prioritize where to innovate, you want to have the agility to stay ahead of the curve, but you also want to ensure that you have people that will actually evolve with this space and not have silos, which is something we see so often in this industry. It's a team sport, as they say. So I'm going to share with you my cheat sheet that I've had for some time, constantly evolves because I got to evolve too, of what I think the pharma of tomorrow will look like. And then I'll talk about where does recursion fit in that? So you won't be surprised to see. Pipeline, whether it's an internal or external pipeline with partnerships, more comprehensive understanding of the biology, supremely important. You can't not do that. This is one of the biggest reasons why we have such high failure rates. Second, novel molecules, but I want to put an asterisk on this. A lot of people are designing molecules that look really cool, but you actually have to have ones that have the right PK/PD profile so that you don't put it in the clinic and then it fails. This happens a lot because people rush things to the clinic. So quality, and that's something that's always been a focus, quality is going to be paramount, more important than speed and cost. Precision development, oh my gosh, how long have we talked about precision medicine? I feel like forever. But if you understand the biology better, you can actually do precision medicine better. And trial design and execution, sometimes people think of it as an afterthought, but 70% of the dollars to make a drug actually goes in development, cost and time. As important as the output is the how, which is the foundation. So I have a bunch here. But if you look at a pharma today, the wet lab is there, the dry lab isn't. Cutting edge AI, machine learning, a few do, slightly biased. High quality data at scale, proprietary data. I think Chris brought up this point that, commercially available data, you hit a limit at some point. You hit a limit. And the reality is, most of the biology, we understand scattered little aspects of it. You need to connect it, because we are a connected system, right? You can't just solve a problem in a specific point. So that is something that doesn't exist. And this is one of the reasons why on that S curve, sometimes people will ask me, there's been so much investment. Why isn't this point higher up in the curve? Because in order to reach that tipping point, you need a huge amount of investment to get the data right? To do the right experiments, to make sure that it's actually generalizable. It's not easy to do. And of course, the little thing around world-class compute. But I will say that the pharma of tomorrow will have many elements that are different, but the one thing that's going to remain the same is to actually make transformational medicines. That word transformational, underlined, because access, adoption, pricing, I didn't talk about the commercialization part, is getting harder and harder, and the bar is getting higher. So now, where does recursion fit in that today? So I'll share with you a little bit of my cheat sheet on that. Look, recursion started in the drug discovery space. Makes sense. Huge focus on phenomics, and then building on that with transcriptomic. I really like the examples of triangulating and being able to validate the dry and the wet lab. That's really great to see. Digital in vivo, I call it the digital endpoints for animals. Look, if you have more sensitivity in your animal models, but there's much more that can be done with organoids and other human models as well, which I know there's thinking around that. Chris and I have known each other for some time. And I remember even 18 months ago, if you looked at this map in my cheat sheet for recursion, it was very dark in the therapeutic hypothesis space and some of the foundations. But very quickly, the company has evolved from molecular invention, I call it generation 1.0, which is repurposed molecules, to molecules that are made in recursion. That is extremely important because one thing that I've seen here and I've been pleased to see is the maniacal focus on actually making medicines, creating drugs. But you can only do it so well unless you develop the most important value asset in any pharma, tech, bio, we can call it whatever we want, company, which is the molecule. The other aspect of it that I think is important, and the team touched on it, is data and AI software. How do you actually democratize this new way of drug discovery across the industry, right? And then more and more work that's being done, and I think this is an area that I'll definitely be leaning into, is how do we go deeper in the discovery space, and then also how do we ensure the emerging work that's being done, leveraging these innovative approaches in development, are scaled. But let me give you another page of my cheat sheet. So as I've been having conversations with various folks in Recursion, I think about some emerging proof points, because that's what makes things compelling for me. I like specificity. So we talk about novel biology that nobody has done before. You heard some of the examples from RBM39, but there's over a dozen examples that Laura and the team are working on in the early and late discovery space. For the size of the team, if you look at industry standards, that's quite a lot. So efficiency is something that you're starting to see. And it's not just for the internal pipeline. It's also for the external partnerships with Roche and Bayer, which I'm sure Matt can touch on more. But it's not just good examples in novel biology in the therapeutic hypothesis space, but also making molecules. All of these programs that I just mentioned, over a dozen, they have molecules that have been made in Recursion. I think that's really important, and we'll talk a little bit about how. But 60% of them use Gen AI. I know a lot of you will probably ask me, like, why isn't it 100%? Because you use technology where you need it, not just because you have to, right? And then the other aspect that was quite compelling to me is, I think it was over the last 4 or 5 years, looking at the speed and the rate being 3x faster, 2.5 times lower, and elevating the quality of the programs. That's where you cannot do that without industrialized workflows. You cannot do that with the scale. You cannot do that without the proprietary data that's been built up over the last few years. Precision development, 7 programs, 5 in the clinic. And I'll talk a little bit about one of the examples where the team is actually focused on doing recruitment in a very different way, orders of magnitude faster than you could do traditionally. But in order to do all of this, and I get this question a lot too, is there going to be one algorithm that can take care of everything? Not yet. But it's important to stitch together multiple aspects that's needed, that's really important to get to a drug. So Phenom 1 around phenomics. What I really like is actually on the chemistry front, having different algorithms for hit ID, for ADME, which I can't emphasize enough the need for that, but also for lead optimization. Lead optimization is probably one of the biggest things that the industry gets hung up on. Super important to do that better. And then Low, which actually integrates, and I'm sure some of you saw it over the demos, integrates a lot of these workflows. So if you look at a lot of pharma companies even today, the data is being generated, not in as a systematic way. Many algorithms are being developed, but it's really hard to bring it all together and to make sure that scientists are actually using it in a systematic approach. I won't go through the rest in terms of compute, but I'm just going to click through some of these examples. So RBM39, and you heard a lot about it, yes, the biology, it's not just that the biology is new, but having a deeper understanding of patient selection is going to set you up for success in the clinic. But I want to reinforce something that Laura said. The 18 months is excellent. But if you look at the number of molecules that were made, I think you said under 1,000, right, 955. I mean, in industry generally, you'd make thousands of molecules. So the waste that goes through it, because you're searching, right? You're searching, and it's not the most efficient way of searching. I think that's something to emphasize. That's where you see the efficiency gain. And then you've seen some of these stats before, but again, I mean, if I look at the size of the teams that are working on this, it's a fraction of what you see in industry. So I think that is important to emphasize. We talk about scale, we talk about efficiency, we talk about having better probability of success. All of that investment is what's needed to actually get it there. And now we need to focus on the quality of the molecules to ensure what gets in the clinic does really well. 2 more examples for me. I really like this one. First, because having been someone in the pharma industry, hundreds of programs, so many programs get delayed, and seeing how you can change that, it's not just impressive to see that you're doing it from 4 to 6 months, and this is for the Axon 1 program, APC, and you can recruit that cohort, and the team was able to in 4 to 6 weeks. But if you just look at the image, today we usually start the approaches. You start by asking the sites, how many patients do you have that fit this profile? And then you go back and forth and you do a little dance, and I've done the analysis, the success rate, like the realization of that is very low. Anyways, and you do all that and then finally get the patients. But in this case, in this approach where you actually use genomics data, real-world data, AI, to actually figure out in this country, where can I find in an anonymized, de-identified way, where can I find the eligible patient populations, where do they exist? And then go recruit them. It's much faster, it's much smarter, and quite frankly, it is the heart of patient centricity, which we've talked about for so long. So watch this to be an area that gets scaled. All right. And look, to do all of this, and I know a version of the slide was shown before, but as I was having conversations with folks, for me, my brain works in discovery, development, commercialization, so I had to map it. And I think it's very important to see here that it's not just building modules and algorithms for the sake of it, but it's actually building the ones that matter. Because if you don't have phenomics, then you're not able to predict some of the off-target effects early on. You're not able to find new targets. If you don't have the transcriptomic, you don't see the full picture of those maps, right, that we talked about, the different layers. And DMPK, digital tolerability, matchmaker, all of those modules are important to get to the right molecule. So I just want to say that it's really, really important to have a collection of these algorithms. And they're not static. They're constantly evolving and getting better. And that's where the agility becomes very, very important. Because there's going to be new modules outside the 4 walls of Recursion. It's going to be very important to be open-minded and opportunistic to bring in what makes sense. All right. With that, I'm going to skip the phenome beta, but it's just great to see that some of these algorithms are actually being leveraged to shape the industry on the outside, not just keeping it inside. So what's next? Look, I think it's going to be really important to be careful and pragmatic as to where we go deeper. So a pipeline that's primarily based on molecules that are using generative AI and other approaches, but also the aspect around clinical development, right? Precision medicine, real-world data, accelerating our trial execution. So with that, I just want to close by saying it's been a great few weeks getting to know the organization. I'm so excited to join in a couple of weeks. And I will say that, look, it's not going to be who has the best and the biggest data set. It's not going to be who has the best algorithm. It's going to be the person or the organization that can actually stitch it all together in a way to drive outcomes, which is about medicines. And I think that that's the ultimate focus that we all need to have. And it's exciting to see that here at Download Day and beyond with the teams. So thank you again.